Good to be here tonight. How many in the audience count themselves as molecular biologists? Just show of hands here. I want to see what I'm up against. Okay, well, I promise not to make you molecular biologists tonight, but we are going to see some amazing things from molecular biology as we seek to answer the question why evolution is not a candidate for the complexity of design we see in nature. I come to this from two disciplines, molecular biology, in which I have a PhD, and in geology, which I have operated as a professional geologist now for 40 years. So I have expertise in both areas, at least some level of it. And so I tried to meld these two fields together to try to tell where it would take us in terms of origin. And this is the result of that study for many years. This animal right here is called a trilobite. Trilobites are very complex creatures. They have a head up here at this end. They have a tail down there, so they are cephalized. They have a complex nervous system. They have compound eyes. And they have swimmerets under their body for doing this and for breathing. And they even have antennae that come out the front of the head. So they're as complex as any modern arthropod, the group to which they belong. So what do trilobites tell us about our history? Well, trilobites are among the first organisms down where this dotted line is, right across the slide there. That dotted line separates the Phanerozoic, that's the time during which we have complex animal fossils, from the Precambrian, where there are no complex animal fossils present. So this is a major milestone in the history of life on the Earth, regardless of how one wants to view it. Below that line, there are no data. Above the line, we have virtually, in the very short period above that line, even in the lowermost Cambrian, we have representatives of every modern group of organisms, every modern phylum. So you can see this is a profound change from nothing below the line to everything above the line. Now, everything, let me qualify that. It means members of every modern phylum. It doesn't mean humans are there, at least that we have any evidence of at this point. But among the creatures that we have is this fascinating one called the trilobite. Now, you may not think it's so fascinating, but there are many trilobite aficionados in the world, people that collect these bugs and play with them. But I can tell you, not everybody thinks that way. One time I was at a store that was going out of business, and they had a particularly elaborate slab of these things. And so I thought, I can't pass that up. It was on sale for less than half price. So I bought it, and I took it home, and I showed my wife. I said, I'd like to put it over there on the wall. And she said, get that out of here. It looks like cockroaches. So I understand that not everybody appreciates trilobites, but I do. And they're fascinating creatures. And they are the first thing we find in the fossil record, right down here at this dotted line. So if we knew something about the molecular biology of the trilobite, then we would know just how complex organisms were when they first occur in the fossil record. So if we had a trilobite alive today, we could study its molecular biology and find out what's going on in there. We don't. They are extinct, and they only go up about that far in the fossil record. But using the tools of modern molecular systematics, we can reconstruct piece by piece just how complex a cell in a trilobite was. And then we can ask the question, since this is the first animal fossil that we find in the fossil record, whatever complexity we can attribute to it has to have come from this black box down here where there are no data. And so the way we're going to do that is we'll look at a fruit fly, a modern arthropod, the same group that trilobites belong to. We'll look at a modern fruit fly, and we'll look at the complexity of the cells of a fruit fly and compare that with the complexity of, say, a human being. If we find the same complex process in a fruit fly and in a human being, then we can attribute that to the common ancestor down here in the Precambrian, according to modern molecular systematics, evolutionary systematics. So using evolution's own tools, we can push the complexity that a modern human and a fruit fly share clear back here into the Precambrian. And if it's in the Precambrian, then we know it's present in the trilobite, the first organism that we find in the fossil record. So we can then answer the question, just how complex was the cell of a trilobite by using this kind of logic. Now you might say, why do you use that logic? Because it forces the question, if evolution is true, what are the consequences in terms of this complexity that we're going to see? Now again, those of you who are not molecular biologists, who are not even interested in molecular biology, you don't have to understand all the details I'm covering tonight, because the issue is very clear. You will see, no matter where you are in terms of your understanding of molecular biology, you will see that what I'm saying is correct. You will see for yourself that the attributes of the fruit fly up here and the human over there that they share in common will go back to this ancestor down here, and that will go up to the trilobite, the first organism we find in the fossil record. All right, so let's start our journey here. But before we do, I want to read a quotation from a modern molecular biologist, Bruce Alberts. He was over here in Amsterdam for a number of years, heading up a think tank on molecular biology. He was head of the American National Academy of Sciences for 8 years, I believe. But he's a very well-known molecular biologist. He's one of the authors on the main textbook that people use to study molecular biology. And here's what he says. We have always underestimated cells. Darwin thought a cell was just a bag of fluid. We have always underestimated cells. The entire cell can be viewed as a factory that contains an elaborate network of interlocking assembly lines, each of which is composed of a set of large protein machines. Do you see the typology he's imposing on the cell here is that of a modern industrial society with machines and assembly lines and factories and motors. Why do we call these large protein assemblies that underlie cell function protein machines? Precisely because, like machines invented by humans to deal efficiently with macroscopic world, these protein assemblies contain highly coordinated moving parts. Now what we're going to do is we're going to start with a very simple example, and we're going to study it for a few minutes, and then we're going to go to a more complex example, and then we're going to go to a more complex example, and until hopefully your head's swimming a little bit. And if you are not interested in all the details, that's okay. Just follow along with the logic. So let's begin our journey with a very simple molecule complex. This is ATP synthase. ATP synthase is responsible for all the energy needs of your body. Essentially, all the energy that makes your muscles contract and your brain function and your eyes work and your ears to hear. All of that, those processes are run by ATP. And this is the machine that makes ATP. It's made up of a dozen proteins. It's made up of a dozen kinds of proteins, several copies of some of them. Here you can see it has down here, the green part is the commutator, the brushes that transfer the energy making stuff onto the commutator, the part that's turning around. And the commutator is turning just like it does in the motor. It turns a shaft, the green part here, up in the middle. And that shaft rotates in amongst those big protein molecules up above. It also has a stator over here, the red protein, that anchors the top part so it can't turn with respect to the bottom. That way, as the shaft turns, something's going to happen up in that top part. It is, in every sense, an electrical motor. In fact, a very clever experiment was done some years ago in which the upper part of this is, by the way, this is the membrane of your mitochondria. The mitochondria is called the powerhouse of the cell. The ATP synthase complex is the motor that makes the ATP, and it's in that membrane. So we're going to take this upper part off and turn it upside down and glue it to a slide. This is a glass slide down here at the bottom. And up at the top, you see that long molecule right there. That's a molecule that's been added to this machine. It's a molecule of actin, the largest protein in your body, just so we can see it. And then there are some dye molecules out there at the end that are attached to that actin molecule so we can visualize it in the microscope, because this whole thing is unimaginably small, much too small to be able to be seen in a microscope slide by itself. So that's what we're going to do, and you're going to see that in this next little video. This is that turning around and around and around. And the reason it's turning is because we have added ATP to it. So just like with a generator, if you turn the generator, it makes power. If you power the generator, it becomes a motor. A motor and generator are interchangeable, and that's exactly the case for the ATP synthase. Now, in the next slide, I want to give you a little bit of preparation so you'll know what to look for. In the next slide, you're going to see outside the membrane, outside the mitochondrial membrane, is a gradient of hydrogen ions. There are lots of hydrogen ions outside, and there are very few inside. So what do those hydrogen ions that are outside want to do? They want to get inside, that's right. And their desire to get inside, believe it or not, this proton gradient, this hydrogen ion gradient, is what keeps you alive. Their desire to get back inside, where there are not very many, is what turns the motor, and turning the motor is what makes the ATP. So you're going to see a bunch of hydrogen ions, and they can hop onto the commutator on one side, and then they travel around inside the membrane to the other side where they can get it back inside. So somehow, it's almost like they know that if they get on over here and go around here and get off over here, they get inside. And it's like they really want to get inside. So that's what turns the motor, and then that turns the shaft, and then when the shaft turns, it structurally changes those proteins on the top so that they force ADP and inorganic phosphate together to make an ATP. You're going to see this all happen. This is the molecule that keeps you alive, so this is something you ought to care about. Here are the protons down here, the hydrogen ions. They're jumping on over here. They get off over here on the other side, and as they go around, they turn the shaft. And when the shaft turns, it takes inorganic phosphate and ADP and pushes them together to make an ATP, adenosine triphosphate. And that's the stuff that makes your muscles work and everything else. So there you are, making the stuff that keeps you alive from moment to moment. How important is this molecule? Well, when people are killed by the gas chamber, what you do is you put some cyanide gas in there and it stops the formation of ATP. And that's how fast you die. So it's very, very essential to your integrity of your body. Here's a modern space-filling model of the same thing. You can see the commutator down here rotating at the bottom. The shaft is going up here, and here you see the ATP coming out and the ADP going in. That's pretty impressive. So now, going back to our diagram and our thinking on this, that molecule, ATP synthase, that whole complex of proteins is present in every living organism, including the trilobite. It's present in all these things. Every living organism, even bacterial cells, have it in their outer cell membrane, and it's what's responsible for keeping them alive. So in this case, we know that it was present down here. Therefore, it was present in the trilobite, the first organism we find in the fossil record. So now we can see there's something really complex in the cell we didn't know about before. And now we're going to take that to another level. You ready? You understand the logic? Is this getting across to you? Okay, so I want to make sure that I don't just leave you in the dust. The point is that that complex structure that we just saw that's in the membrane of every living cell was there in the first cell and there in the trilobite. Therefore, that cell could not have been simple. It had to be very complex. All right, here we go. Let's take a look at cell division. This is a human chromosome. Right here, it has a kinetochore in the middle. The kinetochore is a very complex structure that has a lot of different proteins on it, and it's responsible for anchoring these microtubules that come at those chromosomes from both sides. Now, let's take a look at a human karyotype. A karyotype is just a fancy word for a picture of what your chromosomes look like. Here you see there are 22 different kinds of chromosomes. Let's just take a look at number 1 up there. It consists of 2 chromosomes. One you got from your mother and one you got from your father. So those 2 chromosomes of each type came from both of your parents. Your mother gave you one, your father gave you the other. And now, because this cell is getting ready to divide, these are all doubled so that they can send one of these to each offspring cell. And here is the kinetochore right there. Now, let's talk about the DNA in these chromosomes. Now, remember, these are inside a nucleus of a cell. That nucleus is 10 millionths of a meter across. 10 microns across. That's pretty small. But the DNA in there is huge. Here is one of those chromosomes that has been treated with detergent, and it has released this cloud of DNA that you see surrounding it. And this little box is blown up over here, and you can actually see the strands of DNA going out from that site. So that's a lot of DNA. In fact, even though these are inside 10 millionths of a meter space, some of these chromosomes have up to 16 centimeters of DNA. And when you take the whole set of chromosomes, 2 of each type, you get 2 meters of DNA if you add them end to end. 2 meters of DNA. And when the cell divides, there are 4 meters of DNA in there because all that DNA has to be copied. And copying the DNA is something we'll take a look at in a minute. There's so much DNA in those chromosomes that if you were to look at the nucleus as the size of the Earth, just to give you an analog, make the nucleus the size of the Earth, the DNA in a single nucleus would go to the sun and back 8 times. Another analogy is if you had the DNA, all the DNA, if the nucleus was the size of a shoebox, something we can imagine a little easier than the sun or the Earth, there would be 2,000 kilometers of DNA of kite string in the shoebox. So if you made the kite string to be DNA, you have enough, you'd have 2,000 kilometers of kite string inside a shoebox. That's how much DNA is packed into the nucleus of every cell. Now, that's not trivial because the cell has to keep track of all this DNA. It has to know where it is. It has to be able to divide it up so it makes 2 identical packages when the cell divides. And we'll take a little look at that. Here's a visualization of how DNA gets copied. This is a machine called the DNA-dependent DNA polymerase. It's an amazing machine. It copies DNA. The 2 strands of DNA are going in opposite directions, one going this way and one going that way. So this strand right here at the bottom, is the regular strand, and it just gets copied straight away. The other strand, however, is going the other direction, so it's backwards. So this machine pulls out loops of that DNA and then copies it backwards so that it makes 2 copies of the DNA. Now, something to reflect on as you watch this amazing process. And this is just about real time. So what you're seeing here is how fast it actually gets copied. What you need to reflect on, I think, is that without this machine, how would you have copied the first DNA molecule? If you're making a cell is only alive if it can make a copy of itself, if it can replicate. It's one of the fundamental properties of life. How are you going to do this process without DNA-dependent DNA polymerase? And that's an amazing thing to think about. Well, how does the cell handle all that DNA? The first thing it has is a cluster of proteins called a histone. There are 5 different kinds of histones, 2 copies of each of those. Actually, 4 of them are in this complex, 2 copies of each. So there are 8 proteins in there. And then it wraps DNA around itself, 1.5 turns of DNA. So it condenses the length of the DNA by a factor of 10 or more, just that one process. Now, these histones are interesting just for the fact that they are the same across the whole spectrum of organisms. That is, a histone from a human being and a histone from a carrot differ only in a couple of amino acids. They're almost identical. So that's something interesting to think about. Now we're going to watch what happens to the DNA as it gets wrapped around these histone proteins. Here's a double-stranded DNA molecule here in the middle of the slide. And now you're going to see a histone molecule drop down onto that. Clunk. Here comes some more. And then it goes together. And it condenses the DNA down by a factor of 10. Then 6 of these come together to make something called a solenoid. And that condenses by at least another order of magnitude. And then that structure, that thread, coils. So you see here. And then that coils again. And then that coils again to make the chromosome. And now you can watch the cell as it divides. Here are these chromosomes getting into the middle of the cell. And if the cell were to divide prematurely before everybody was there, it would die. But it waits until the exact moment that all the participants are in order, and then suddenly on a signal, they separate at the kinetochore and go to opposite sides, half of the chromosomes on one side, half on the other, and now we have 2 cells. A process that is going on in your body trillions of times while you're sitting, or well, billions of times while you're sitting here. So this is a remarkable thing. And all that condensation of DNA that you just saw from a single strand to this coiled coil, it happens without any little men in there. We haven't ever seen any little men in there that are trying to help it out or tell it what to do. It's all designed into the system. So there is a super architecture to the cell that integrates all of this complex process so that it happens simultaneously and it happens without any intervention that we know about. So here we have this set of chromosomes. Now we're going to take another look at that division process in a little more detail. We'll go back to that same structure of the chromosome. This is a little different rendering. This is the work of Drew Barry and his group that have made these videos. By the way, these are animations. These are not the real thing. But here you can see the DNA wrapping around the core of histones right there. And this is all one chromosome that you're seeing here, condensing down and making a structural structure that can be separated and kept track of. Now we're going to draw back out of the nucleus. We'll go through the nuclear pore, which we'll say more about in a minute. And we're going to go back out into the cytoplasm, and then we'll watch that cell divide again. Now think about the process of these chromosomes having to separate at the middle where they're joined on a given signal and then go off to opposite sides in such a fashion that every single chromosome gets half of it, gets one of its copies in one cell, the other copy in the other cell. There they go right now. They've just separated. Now they're going to opposite sides of the cell. And if it makes a mistake, if one of those chromosomes gets left behind, then they all die. And that's not a good thing to happen. And so it has to happen right. Now we're going to go back and pull out one of these chromosomes, right, this one right here, and we're going to study what happens to it. So there you see that one chromosome. It's joined at the kinetochore by the microtubules so that they can be pulled apart when the signal is given. And there's about 2,000 different proteins at the kinetochore. This is a very complex structure in the cell. It requires 2,000 different proteins to be involved in this process. And we're going to ask the question, how does the chromosome know when all the other chromosomes are lined up and ready to go? And we'll look at that by zooming into the kinetochore itself. Here you can see the microtubules connected to the kinetochore, and now we'll zoom in closer, and you can see the microtubules are growing, and they're expanding out, and they're growing away from this kinetochore. And the kinetochore is sensing when it's in the middle of the cell. Somehow we don't know all the mechanisms, but somehow the kinetochore is realizing that everything's just right, and it's now in the middle of the cell, and so it sends out a signal to tell the other chromosomes that it's good to go. And here you can see these things turning green. That's symbolic that they have now recognized it's time to send the signal, and there goes the signals right there. Boom. And they don't just float out in the cytoplasm, they walk. They walk just like you and I walk. This is not made up. Here you can see these microtubules are genuinely highways in the cell. And the molecules that travel from place to place in the cell use these microtubules to walk from place to place. And we'll say more about that a little later. But there go the signals out to the cell telling it it's time to divide. And that's how that process works. Isn't that amazing? See, you don't have to be a molecular biologist to appreciate the complexities of the cell. And this is stuff Darwin didn't know anything about. Let's take a look at one more feature of the nucleus that's involved in the cell division. This is a modern representation of a cell. Here is the nucleus. Notice when you switch from the cell to the nucleus, now the nucleus looks just as complex as the whole cell did. But the nucleus is peppered, the surface of the nucleus is peppered with these nuclear pores. Nuclear pores are extremely important for the function of the nucleus. Remember, the nucleus is where all the information and guidance for the cell comes from. So what gets in and out of that nucleus is essential to the cell. It's essentially fundamental. And the nuclear pore is designed to screen molecules, so it only lets molecules out when they're prepared to meet the environment that they will encounter. And it's designed to keep things out of the nucleus that don't belong there, but let things in that do, and to allow things to get out of the nucleus that need to get out in the cytoplasm. So the pore is very important. But look what happens when a cell divides. What happens to the nucleus when a cell divides? If you've had general biology, you know the nucleus dissolves. And that means the nuclear pores dissolve, and that means the nuclear pores have to reform in the nuclear membrane every time a cell divides. So right now in your body, this is going on. Let's take a look at what's involved. Here is a modern representation of a nuclear pore. This is from a yeast. It has 456 different proteins in the pore of 30 different kinds. Just a nuclear pore has 30 different kinds of proteins, 456 of them total. And so here you see the structure of the nuclear pore coming together in the nuclear membrane, and this is going to make that pore reestablish itself every time the cell divides. These proteins not only have to know what to do, what their function is, what they're supposed to do, but they also have to know how to put themselves together to make the nuclear pore. It's like you had a car. Your car is supposed to take you somewhere, but if you need another car, you don't go to your car and say, okay, make me another car. It's not going to work. You're going to be waiting a long time. A car knows how to take care of its function, but it knows nothing about how to make a new car. This nuclear pore knows how to assemble itself. It also knows how to do the function of a nuclear pore. So that's a profound difference. Okay, all of that complexity that we just elaborated, which is a tiny, tiny fraction of all the complexity in the cell division, all that complexity is present in plants, in humans, in fruit flies, and in every other cell that has a nucleus in it. Bacterial cells are similarly complex, but we won't go into the differences because it's not relevant. But that means if our hypothesis is correct, that means this common ancestor had to already have all that complexity of cell division present in it way back here in the Precambrian, and that means it was present in the trilobite, the first organism we find in the fossil record. All right, let that settle in for a minute. Now we're going to something more complex. Well, what's more complex than cell division? Well, what about the nerve impulse? Let's take a look at that. Here is a afferent neuron. Here is the synapse. Synapses are very important. That's the place where a lot of poisons work. It's a place where psychotropic drugs affect behavior and things like that. This is the neurotransmitter. It's released from the afferent neuron. It travels across this space, which is very tiny, but it's a space nonetheless. It's like an insulation. And then it has receptors on the other side, and these receptors respond to activation by the neurotransmitter and then stimulate the postsynaptic cell. An example, if you touch your finger into a fire on something hot, before you can move that out of the way, this has to happen. And there has to be a transmission across the synapse, and then it goes to a muscle cell, and the muscle cell retracts your hand. So this is very fast and very efficient. So when you have a stimulus here, you can respond very quickly with your muscles through this process. Let's take a look at how it works. This is one of the proteins that's responsible for the nerve impulse passing down the nerve cell. It's called look at the name. That tells you something important's going on here. Voltage-gated sodium channel protein. Voltage-gated, that means it has a gate that's open and closed in response to a change in voltage. It is a channel, and it is a channel for only sodium, and it is a protein. So one protein has to do all that stuff. Well, here it is. Fairly complex protein. This is the cell membrane, and that protein goes through the membrane 24 times. That in itself is a complex process because it has to be synthesized in place. It's synthesized in the membrane, and the amino acids in the protein specify to go into the membrane and come back out again, go into the membrane, come back out. Each one of those passages has to be built into the code of the amino acids in order to get the protein to be synthesized. Once it's synthesized in the membrane, we think it forms some kind of a cylindrical structure, something like this, if you want to imagine it. And what happens is an impulse comes along. Here's a resting pore. Notice that the gate is closed. Here's one here. The gate is closed. Then a pore is open. It can open for from a variety of stimuli, but it's open. And when it opens, it allows sodium ions, which are plus charged, to come inside. Those sodium ions migrate along the inside of the surface to the next pore. They cause that to open. That opens, more sodium comes in. It goes over here and opens this one. And that's how the nerve impulse travels along your nerves as you are doing things or thinking thoughts and all that. All that depolarization event is based on these sodium channel proteins. Now, because the gate is open and it's open by having plus on the inside, you're dead after one impulse if something doesn't change. What changes is this ball and chain here on the inside are attracted back to that pore and they block the flow of sodium ions. They block it so no more sodium can come in. Then the sodium-potassium pumps the sodium back out. The gate closes and it's back in its resting state. Wow, that is complex. That is complex. Eventually, that impulse gets down here to the end membrane of the cell, and it releases this acetylcholine that you see right here, the little red ball. The acetylcholine goes across and stimulates the next cell and causes an effect. Well, let's take a little look at that acetylcholine and how it's treated in the cell. Here you see a representation of the process. Here the acetylcholine has been released to the outside down there at the bottom. Then the little vesicle, these are called vesicles, the vesicle is recaptured, taken back inside the cell, and then it's refilled with acetylcholine so it can be used again, and then it travels back down here to the end membrane and releases its contents again. Well, that's nice, that's interesting, but what keeps it from just diffusing out into the cell? Well, we've already had a little hint as to what that is. Let's take a look. Here is this is actually a photomicrograph, a time-lapse photomicrograph of a microtubule. Now they're really, really tiny, so somehow this has been enhanced. And these are two vesicles, one there and one there, and look what they're doing. They're walking along the microtubule. They're walking along the microtubule from one place to another. They're passing each other, and here they go on their way in opposite directions. These vesicles are actually carried by little men, if you will. They are carried from place to place by these transport molecules walking on the microtubules. And that's what you see right there. Here they're meeting in the middle, maybe exchanging greetings, or how's things down at your end of the cell? We don't really know what they're saying. That was an anthropomorphic slip. Okay, so here you can see an electron micrograph of that same thing, and there's the fiber of the microtubule, and here's two vesicles right in the middle showing you that it really is that. Now, here's a ribbon model of a protein doing, well, sorry, this is just a picture out of a book, and it shows these little men carrying that vesicle from place to place. This is the most sophisticated molecular biology textbook on the market. And they draw a picture with two little men. How can people keep a straight face when they see that? Okay, let's watch how it works. Here's a ribbon model. That's the microtubule right there going across the middle. And here is the kinesin molecule walking on the microtubular proteins from place to place. Here is a space-filling model of the same thing. And notice what it's using to take its steps. ATP that we made in our first study. So here it is walking along the microtubules from place to place. Here is a video made by Howard Hughes Medical Institute for Harvard University. I know some of you have heard of Harvard. And so it's not fake. This is the way it's really thought to work. And here is how they depict it. Here's the vesicle up here. Here's the kinesin molecule, and it's walking along the microtubular highways. It's not just the vesicles that walk on the highway. Other things do as well. Here is a depiction of a mitochondria. Mitochondria is the powerhouse of the cell. This is where the ATP is made. And it's not just floating around inside the cell. It's going to where the energy is needed and dumping off a load of energy. It's like a fuel truck. And look, there it is walking from place to place on the microtubular highway. So all these things that we thought were just happenstance in the cell, when we examine them more closely, it turns out, no, they are highly engineered. They are designed, obviously, by something external to the cell itself to carry out functions that are profound and defy logic if we think they could have happened by accident. This is all a programmed, designed entity. All right, well, let's go look at what happens when these vesicles get to the end membrane. Here's the end membrane of the synapse right here. Here's the pore that releases the contents of the vesicle to the outside on signal. It's anchored right here by some special proteins. But look, what's this over here? It's called the Rab protein. The Rab protein is the shipping label. Now, I don't know who carries shipping in Netherlands, but in the United States, it's UPS. So this is a UPS label, and when it gets to the end membrane, the cell scans it right over there on that side. The cell scans that label, and if it doesn't say end membrane of the cell, then it turns it around and ships it back out in the opposite direction. So there is a whole postal system inside the cell that's designed to make sure the right vesicles end up at the right place. Not only are they carried there, as you can see up here on the microtubules, or spectrin in this case, not only are they carried there by little men, I don't know what you want to call them, little protein molecules that are walking, and brought down to the membrane, but once they get there, then they get checked by the UPS guy to make sure that's where they belong. And that's profound. In the cell, there are highways. There are motor transports. There are shipping departments. There are delivery systems. There's postal systems. All this kind of stuff that we think of in our world that makes up our world, we're finding are inside the cell. So in reality, the cell itself is more complex than the organism from which it's made, which it composes, sorry. One more thing, and then we'll draw this to a close. This is, now we've got that membrane fused into the end membrane of the cell. We need to get it back because all those specialized proteins will be lost. So we have a molecule in the cell called clathrin. And clathrin forms a cage about that membrane and rescues it and sends it back into the cytoplasm. And here we can see that happening in this case. Here is the vesicle up here just entering into the cytoplasm, and it's going to come through. Notice there's a cytoskeleton here. There's all kinds of structures in the cell. We didn't know about any of that either. But here comes the vesicle surrounded by clathrin. It gets back into the cytoplasm, and now the clathrin falls off and releases the vesicle so it can be filled up again and used used another time. All that complexity of the nervous system and a whole lot more that we could elaborate is present in fruit flies, it's present in humans, and it's present in every other organism that has a nervous system. That means it had to be present in the common ancestor of humans and fruit flies down here in the Precambrian, and that means it was present in the trilobite, the first organism we have in the fossil record. Now these examples I've given you are three examples of 100 that I could give you and maybe 1,000 that I could look up details on that are as complex or more complex than this. And I would love to share some others with you, but time doesn't allow. And besides, I think you're getting the point. So we can easily say that virtually all the complexity of any modern organism, of the cells of any modern organism, was present in the first organism we find in the fossil record, the trilobite. All the complexity of any modern cell was already present in the trilobite, the first organism we find in the fossil record. Even though it's extinct, we can say that with assurance if we believe in the evolutionary hypothesis. That being the case, what does that tell us about the world around us? What does that tell us about where we came from? It means that if we believe that evolution is the origin from which we came, it means that all that complexity had to happen down here in the Precambrian where there are no data. So we believe, if we're evolutionists, we believe that all this complexity took place down here in the Precambrian where there are no metazoans for it to have happened on. And when this first organism shows up in the fossil record, it's already got all that complexity. Anything else we would propose would be eliminated, because that's the basis of all modern systematic taxonomy, molecular taxonomy. So if you believe in evolution, you believe it on the basis of a very poorly defined faith. You believe that all that complexity arose below this line without any data to support it. And if you're a creationist, you believe that God created things, at least you have an explanation that makes sense. Now, you might want to argue about where God came from or something else, but if you have an all-powerful God, if you visualize God as being an all-powerful God, He could make something this complex. Otherwise, He's not all-powerful. And His fingerprint is in it, and you can't get it out. But if you believe it happened without a God, it happened just by chance, then you're committing yourself to a belief in something strictly on the basis of blind faith. Not even faith, it's blind faith. And I'll leave it at that. Thank you very much.